What if we could detect cancer from a simple blood test? No surgery, no biopsy. Welcome to the world of liquid biopsy. Liquid biopsy is the future of cancer care. Faster, safer, smarter. Liquid biopsy is changing the way we detect and fight cancer. Are you ready for the future of medicine? Liquid biopsy is a minimally invasive technique that analyzes tumor-derived material circulating in body fluids, most commonly blood. We can use any type of body fluids. Unlike traditional tissue biopsy, it is safer, repeatable, and can capture the genetic diversity of tumors throughout the body. Circulating tumor cells or CTCs are cancer cells that break away from the primary tumor and enter the bloodstream. To detect them, scientists look for specific markers. These include EpCAM or epithelial cell adhesion molecule and cytokeratins, that is CK8, CK18 or 19. At the same time, CD45, a leukocyte marker, is excluded to make sure normal white blood cells are not counted as tumor cells. CTCs are found in several cancers including breast, prostate, colorectal and lung cancers. There are multiple methods to isolate and analyze. The cell search system is an FDA approved for clinical use, while microfluidics based capture and immunomagnetic separation are widely used in research and allow precise detection and enrichment of circulating tumor cells from blood samples. Next is circulating tumor DNAs, that is ctDNA. It consisting of small fragments of DNA that is released into the bloodstream from tumor cells undergoing apoptosis or necrosis. These DNA fragments carry the genetic information of the tumor, including specific mutation or epigenetic changes. Some of the commonly analyzed mutations include EGFR, KRAS, BRAF, TP53, etc. In addition, certain methylation patterns like SEPT9 in colorectal cancers can also indicate the presence of a tumor. ctDNA is particularly useful in detecting cancers affecting lungs, colorectal cancers, breast and pancreatic cancers. To analyze ctDNA, highly sensitive techniques are used. These include PCR-based methods such as digital droplet PCR or quantitative PCR, which allow detection of even tiny amount of mutated DNA. Next generation sequencing or NGS provide a broader view of the tumor's genetic landscape. Another method called beaming or beads, emulsion amplification magnetics, combines PCR and flow cytometry to precisely detect rare mutant DNA fragments. By studying ctDNA, clinicians can not only detect the presence of cancer but also monitor disease progression, detect minimal residual disease and track response to therapy, all from a simple blood sample. Did you know that every cell in our body releases tiny particles called extracellular vesicles or EVs? Among these, exosomes are the smallest messengers packed with DNA, RNA and proteins. Tumor cells release EVs in high numbers, carrying clues about cancer. For example, glypican-1 is linked to pancreatic cancer, while certain microRNAs like microR21, ON41 and tumor antigens such as HER2 and EGFR can all be found inside. Scientists use methods like ultracentrifugation, size exclusion, immunoaffinity capture, and nanoscale flow cytometry to detect and study these vesicles. Exosomes offers a non-invasive way to detect, monitor, and even target tumor, a tiny particle with a huge impact. Next is circulating RNA or microRNAs. These are small non-coding RNAs. They are usually short RNA molecules that do not code for proteins but play a critical role in regulating gene expression. In cancers, certain microRNAs are released by the tumor cell into the bloodstream, often packaged with the exosomes or bound to protein complexes. These packaging protect them from degradation, making them highly stable and reliable marker for liquid biopsy. Different microRNAs are associated with specific cancers. For instance, microRNA 21 is commonly elevated in breast, lung and gastric cancers, while microRNA 141 is frequently associated with prostate cancer. These microRNAs reflect the biology of tumor, including its growth, invasion and metastatic potential. To detect and analyze these circulating microRNAs, researchers employ sensitive molecular techniques such as RT-PCR, which allow precise quantification of specific microRNA. High-throughput RNA sequencing can also be used to profile the entire spectrum of microRNAs in a sample, providing a comprehensive view of tumor-associated RNA patterns. By studying these small non-coding RNAs in blood, scientists and clinicians gain a powerful non-invasive tool to detect, monitor and understand cancers, potentially improving early diagnosis and guiding personalized therapy. Next, let's explore tumor-educated platelets or TEPs, a fascinating and emerging tool in cancer diagnostics. Platelets are small enucleate cell fragments primarily known for their role in blood clotting. However, recent research has revealed that they can also interact with the tumor cells in a unique way. When platelets are exposed to tumor-derived signals, their RNA content changes, effectively educating them. These tumor-educated platelets carry tumor-specific messenger RNAs or mRNAs which reflect the presence and type of cancer. By analyzing these RNA signatures, we can gain insight into the molecular profile of the tumor without directly sampling the tumor tissue itself. Tumor-educated platelets have shown promise in detecting a variety of cancers, including lung, pancreatic, and colorectal cancers. The primary method for analyzing these platelets is RNA sequencing, which allows for high-resolution detection of tumor-specific RNA changes. In summary, tumor-educated platelet represents a non-invasive, highly sensitive approach to cancer detection, offering a window into tumor biology through a simple blood sample. Their ability to mirror tumor-specific RNA changes make them a powerful tool in the emerging field of liquid biopsy. How the liquid biopsy works. I already told liquid biopsy is an innovative, minimally invasive approach for detecting and monitoring cancer through body fluids. So the process begins with the sample collection, typically blood around 5 to 10 milliliters, though urine or other body fluids can also be used. These samples contain tumor-derived components that carry vital information about the cancers. So once collected, specific tumor components are isolated for analysis. Second step is isolation of the tumor-derived components. Circulating tumor cells or CTC can be enriched using antibodies, targeted cell surface markers or by size-based filtration methods. Cell-free DNA or cfDNA is extracted from plasma after separating it from the cellular components. Exosomes, small vesicles released by the tumor cells, are isolated using ultracentrifugation or specific immunocapture techniques. So, after isolation, these components undergo detailed molecular analysis. The DNA mutation are detected using PCR and next-generation sequencing, providing insight into the tumor-specific genetic alterations. RNA profiling using quantitative PCR or sequencing reveals gene expression patterns. Also, protein markers can be assessed through methods like ELISA or flow cytometry. Finally, the collected data are interpreted to assess the presence or absence of a tumor, identify specific genetic mutation for personalized therapy guidance, and monitor disease progression or minimal residual disease. This comprehensive approach allows clinicians to track cancer dynamically, offering a powerful tool for precision medicine.